Thanks so much, Matt. As Matt mentioned, I'm Jen Herbach. I'm one of the co-founders and the CEO of Adventris. Really excited to be back here today. I want to thank Matt and the entire JHTV team for having us back here. So before I dig in, I just wanted to as Matt mentioned, we were here last year and we have a lot of exciting updates to share. Particularly, I think when we were speaking here last year, we were really focused on being what I would call a best-in-class antigen company and saying, how do we make the best cancer vaccine antigens? And I think one of the things that has really changed between now and last year is we're now really focusing on how do we make the best possible cancer vaccine. So really excited to talk you through our updated story and also introduce you to our expanded management team that we have grown since last year. So when we think about Adventris, I think there's a couple key things that I want to talk about to begin with. The first really is why now for cancer vaccines, and then why us? So when we think about cancer vaccines, the randomized phase 2 data from Moderna in their personalized cancer vaccine in melanoma has really reinvigorated the field. And it was really exciting because this was the first randomized trial that incorporated all the key learnings that we've seen in the cancer vaccine landscape over the past 10 plus years, including it's really important to go in the early stage disease. It's important to target multiple tumor-specific antigens. And then additionally, the strength of the T-cell response matters. And this is probably one of the most important key insights that we have seen. And we've seen this across multiple trials, multiple platforms, multiple tumor types, that if we get immune responses above a certain threshold, we see that translate into clinical benefit in the adjuvant setting. And then finally, now with the ability to combine with checkpoint inhibitors to prevent D-cell exhaustion. So when we think about Adventris, what we're really focused on is how do we take all of these key learnings but apply them to an off-the-shelf vaccine? Because when you think about a personalized versus an off-the-shelf vaccine, from the complexities, an off-the-shelf vaccine will win. So then the challenge becomes, how do we apply this? So first, we're going to be going in the neoadjuvant setting or early disease setting. And most importantly, when we think about our special sauce, and I'll talk about this a little bit more, is we are targeting KRAS, which is the most common oncogene. And we have developed what we call our antigen engineering platform, which enables us to solve the problem of going after these antigens which are not very immunogenic. And then in addition, we're using very potent vectors to maximize the immune response, and then finally combining with the checkpoint. And then I'll talk a little bit more about the end, about our path to the clinic, but most importantly, we're targeting dosing our first patient end of next year. So not surprisingly, since we're here, we're a spin out from Johns Hopkins. And the Hopkins academic team did what we call the first generation KRAS vaccine trials. And this was one of the very earliest trials targeting KRAS. And there were a couple of key learnings that came out of this trial that really led to the formation of Adventris. The first being the strength of the immune response matters. In addition, the second being that KRAS is not naturally immunogenic, especially KRAS G12D. So we were really founded to solve these two issues of saying, how do we improve the immune response to KRAS and improve it for the majority of patients so we can see clinical benefit for those patients? And when we think about how to do this, there's really the two key components of vaccine immunogenicity, the antigen and the vector. And we have designed our approach to maximize both. So as I mentioned, what we call our lead candidate is what we call our engineered KRAS plus vaccine. And there's two key components that I want to address. The first is we're including what we call our engineered KRAS antigens, which solve the problem of being able to target this challenging antigen. And then we're also including additional shared tumor-specific antigens to just drive the response. And then additionally, we've chosen a best-in-class vector, and we're going via heterologous prime boost system, which I'll talk a little bit more about shortly. So I'm not going to spend much time on this slide, but I think as everyone here probably knows, KRAS is the most common oncogene. It's present in pancreatic, colon, and lung cancer. Our lead indication is pancreatic, where it's present in pretty much almost all patients. And KRAS G12D, which is the most common KRAS mutation, is also not surprisingly the least immunogenic. And we can see here, this is data from the Hopkins first generation trial, that if you look at the bottom, KRAS G12D is the least immunogenic. And all the patients with a KRAS G12D antigen reoccurred in the trial. So the question then becomes is how do we improve the immunogenicity for KRAS G12D to be able to target all of these patients? And we have engineered KRAS G12D to substantially improve it. And I'll show you that data very shortly. So before I go into explain a little bit about our platform, I just kind of wanted to take a very high level step back. So if you look on the, I guess, right of the slide, you can see that this is the MHC binding groove, and then you see a minimal epitope that is bound in there. And so the red residues are basically what we call the anchor residues, and those really determine the immunogenicity of a target. Whereas the yellow residues, those are the TCR contact residues, and those determine the specificity. And one of the things that we can do is we can actually separate those two functions. So we can make modifications to the anchor residues so we improve the immunogenicity of the target without actually compromising the specificity. And so this is obviously a little bit of an oversimplification of what we do, but at a very high level, we can separate these two functions. So we can improve the immunogenicity of KRAS without actually compromising the specificity to KRAS G12D, for example. So for the immunologist in the room, this is basically the principle of molecular mimicry, where we're giving the immune system something slightly different and tricking it into recognizing what we want it to recognize. So if you look on the top of the slide, this is an example of kind of what all the other KRAS vaccines have done. You've basically given the exact same native sequence that is in the tumor. And then the challenge has become is that you're giving the immune system something that it already didn't recognize because it developed that KRAS-driven tumor to begin with. And what we're doing and what's so unique is we actually make specific amino acid modifications to these KRAS sequences and give the immune system something slightly different to actually maximize and drive immunogenicity. So this is an example of one of our engineered KRAS G12D antigens. So if you look on the bottom, that basically green bar, that's basically what everyone else has done. And this is kind of the natural immunogenicity of KRAS G12D. Whereas you look at the purple bar, that is an example of our engineered KRAS G12D antigen. And you can see it's significantly more immunogenic. And if you remember at the beginning, I said that one of the key takeaways that we've seen in all of these trials is that the strength of the immune response matters. So if we can get patients above a certain immunological threshold, we see that translate into clinical benefit. So we have generated a pool of these KRAS G12D engineered antigens to drive the immunogenicity for these patients. So as I mentioned at the beginning, there's kind of two key components when you think about immunogenicity of a vaccine. The first is the antigens, and the second is the vector. And so when we think about our vector delivery system, our goal is really how do we deliver this vaccine in a way that maximizes the strength of the T cell response. So how we're approaching this is we're delivering it via a prime boost system where we're priming with peptides. And we know that peptides are really good at maximizing CD4 T cells. And then we're going to be boosting with an amplifying RNA. And we know that these are very good at maximizing CD8 T cells with the goal that by combining these two, we can maximize both CD4 and CD8 T cells. In terms of our clinical trial plan, as I mentioned, we're planning to dose our first patient end of next year. We're targeting a phase 1 in the adjuvant setting in patients with resected pancreatic cancer, and then have a very clear go, no-go decision based on the fact that we have all of this data from 60 plus patients in the first generation trial showing the immunogenicity levels of those patients and to really have a nice proof of concept if our approach is working. And then from there, go very quickly into a randomized phase 2 with really our focus on how do we get to very clear randomized phase 2 data as quickly as possible. As I mentioned at the beginning, and I'll be joined shortly by my colleagues, we've really been able to build out our team since last year. Dr. Mark Yarchoan, who is all this work kind of stems from his lab, is our CSO and co-founder. We've also brought on Andrew Allen, who's the former CEO and co-founder of Gritstone. And then none of this would be possible without kind of all the leadership of Dr. Liz Jaffe, who is one of our scientific advisors and co-founders. We've raised, in terms of where we are right now, we've raised a double-digit seed, and we'll be going out shortly to raise our Series A. From a timeline perspective, we're planning to select our development candidate at the end of this year, and then targeting dosing our first patient at the end of next year. And now I'll open it up for questions and join.
Thank you, Jen.
Hey, thanks for the presentation. Quick question about the HLA optimization piece. How do you guarantee, I guess, that the binding is going to work across, I guess, multiple HLA alleles across a broader population?
Is this on? Oh,
Yeah.
Good. Yeah, that's a great question. I'll answer this briefly, and then we can chat more afterwards. But our screen essentially uses known minimals of KRAS and optimizes for known minimals, which are HLA specific. So we initially started with known minimals like KRAS G12D in the context of A1101 and other minimals, made modifications for those minimals. And so these minimals are often HLA specific. But what we found is, even though our screen was designed to screen them in an HLA-specific way, many of these minimals, when we actually have gone and tested them, have worked in donors who do not have the HLA that we screen these peptides for. And what we've learned is that there are HLA families that have very shared features. And so, for example, our minimal that was screened in a CO8 screen actually works best in a CO5 donor. And so we've learned a lot. We're getting better at this, and many of our minimal epitopes cover a broader population than we had expected.
Thanks, Mark.
Faye.
Thank you. Congratulations for such successful race and moving forward. My question is, so you did a very good job explaining to us you're pushing the signal over a threshold, now allow our T cell to recognize. But I want to ask the other side. What happened so when you do your trials, forward trials, do you stratify patient population that has a TCR able to recognize your G12D?
Sure, maybe I'll take that. Simple answer is no, because we're delivering multiple epitopes, both KRAS, but also many additional epitopes from what's now referred to as the dark matter of the genome. And so there's a transition, I think, that's happening between fully personalized vaccines to an off-the-shelf approach, which shifts the economics dramatically. And as the CEO of a company that worked on personalized, it is prohibitively expensive. In this capital environment, it is simply not possible to raise the 100 million plus you need per year to be able to produce personalized vaccines in a fully GMP compliant manner. And so we have to move to off-the-shelf, predetermined vaccines. The only two companies really still driving on personalized at scale are companies with massive capital. So it's Moderna, Merck, and BioNTech. BioNTech/Roche Genentech. So those are the only two that can still support the fully personalized. So as we move to the off-the-shelf, we've got the KRAS approach. And of course, there will be some patients who are more inclined to respond. As Mark said, we're seeing this evidence that HLA superfamilies are sort of functionally relevant. And so there is a breadth of repertoire that may be adequate to drive a TCR response or a clonal response against a particular KRAS mutation. But we also have these other dark matter antigens which are highly expressed, tumor-specific, and shared. And so this is not relentlessly focused on KRAS. KRAS, I think, is super important, and it's a great anchor for a product, but it's not the entirety of the product. And so we don't over-index on a KRAS-specific response.
One more question.
Yeah, thanks. My question was really on how much of a cash runway for your Series A, and what are the value inflection points you're going to have in Series A?
Yeah, so I'll take that one. So in terms of where we're at right now, we raised a 12 million seed. We have 9 million still left in the bank. So we still have a decent amount of runway. Our plan for our Series A is we're going to plan to fund the company through randomized phase 2 data. So to get it to a very, very clear value inflection point.
Great talk. Thank you. The question I have is, how do you see the impact of the KRAS inhibitors, particularly the G12D from RevMed, which is covalent, and SHOCAT has already shown that, that basically is a post-translational modification, right, when presented?
You should take that.
Yeah, I can take that. I'm a practicing oncologist. I've treated patients with these KRAS drugs. They're amazing, right? The patients have rapid shrinkage of tumor. But every single patient that I've ever seen develops acquired resistance. And, I think this is an age-old question that we get every single time there's an immunotherapy and a targeted therapy. This is not the only time that we've had both. I think I see a role for both in this space. And, I think that vaccines generally have shown the most efficacy in these micrometastatic settings. I think targeted therapies work very well for debulking bulky disease. And in the very long run, it wouldn't surprise me if patients actually end up getting both in various combinations. Obviously, many steps ahead of where we are.
Okay, we're actually right at time for a break, so let's thank Jen and the Adventris team one more time. Thanks.